Alright, so this lecture is going to deal with enzymes, specific proteins. We've already seen making proteins. Enzymes are some of those proteins. Enzymes are going to help catalyze reactions. Okay, many reactions, they would be very, very slow. So if we needed a reaction done, it could be very, very slow. But we have enzymes that allow us to speed up these reactions to allow the cell to be able to operate. Okay, so many of our proteins, many of our genes code for these enzymes. This material is it's bunched in there with cellular metabolism. So I'm gonna chiefly deal this with enzymes. This lecture will deal with enzymes, and then we'll have a separate lecture dealing with cellular metabolism. But both of these lectures will be covered in chapter 4. The material will be in chapter 4. Okay, but chiefly I'm just going to be looking at enzymes here and what their functions is, how they're regulated, some conditions or properties that can affect enzyme activity. Okay, because these are very important. Our body works through chemistry and through chemical reactions, so these enzymes are very, very important for our functioning of our cells. As I said, enzymes. What are enzymes? Enzymes are proteins. Okay, we have genes that code for many, many, many proteins. Okay, and these can do speed up all these different chemical reactions. They are called catalysts. Okay, their function is basically to speed up the rate of reaction, speed up reaction rates. Okay. You have a classic example is maltose. Maltose is 2 glucose molecules bonded together. If you put maltose into water, okay, remember the sugars are broken down by hydrolysis. If you put it in water, what happens is maltose doesn't break down. You can come back 100 years later and much of the maltose is still as maltose. It hasn't split apart into glucose. Okay, so we need this to take place, for digestion to take place, and so that is the function of enzymes. They're going to be able to allow us to speed up this rate of reaction to allow us to break that maltose into 2 glucose molecules. Okay, that's just one example of chemical reaction, but there are all these thousands of chemical reactions that are taking place in our body, and we are using enzymes to make them speed up. Okay, so that's the long version of basically enzymes are there to increase the rate of reaction, right? They are not changed by the reaction. Remember, our reactions are taking a substrate, breaking bonds, and putting bonds back together. Okay, so those molecules are changed. We don't lose any atoms, but the molecules are changed. In the case of the enzyme is not changed in the reaction. Right? This is key because then we don't have to, if they were changed, we couldn't use the enzyme again, right? So the enzymes aren't changed because they can constantly be catalyzing these reactions. Okay, they do not change the nature of the reaction. Okay, if an enzyme catalyzes A, B to C and D, it's not going to catalyze A, B to F and G. Okay, it's not going to change what the substrate or the products are in the reaction. All it simply there is to speed up the reaction, make the reaction go faster. Right? And the way it does this is basically it lowers what we call the activation energy. These chemical reactions there has to be a little bit of energy put in to make these chemical reactions take place. Okay, some of these reactions, the reaction energy is great. For some of them, it might be a little bit smaller, but essentially, basically, what the enzyme doing is lowering the amount of energy needed for the reaction to actually take place. A classic example is a match. Okay, you can look at a match and you're not going to be able to get it lit, right? It's not going to catch fire. But if you just strike it All right, some of those you can just strike gently, and what happens? You added that activation energy. That activation energy allows it to become a flame. And that's simply what these enzymes are doing. They're allowing these reactions to take place by lowering the activation energy. Okay. What is the activation energy? Essentially, energy required for the reactants to engage in a reaction. Okay, many of the molecules that the substrates, okay, the reactants don't have enough energy. Like the case of that maltose. The maltose, there is not enough activation energy to basically cause a splitting of the maltose. Okay. We could do this several ways. We could add heat to make possibly the reaction take place quicker. Okay, heat does increase the rate of reaction. Remember, these solutions in which these chemical reactions are taking place, these molecules are in the solution and they're moving around. They have this what's called kinetic energy. Okay, they don't just sit still. Remember diffusion. Okay, they don't just sit there. They're in the solution and they are bouncing around. If you increase heat, say you have A and B, the likelihood is those can hit together and we can have the chemical reaction. So we can add heat. But the key is right, you saw with proteins, if we add too much heat, proteins start to denature. Okay, we can only raise it so much before it causes problems in the body. So instead of heat, we have our catalysts. Okay. The enzymes allow us to allow those reactions to take place without increasing the need for heat. Okay, and basically again it is to lower the catalyst, lower the activation energy, the energy needed for the reaction to take place. Here's kind of a difference between a catalyzed and a non-catalyzed reaction. Okay, the green are the number of molecules, the number of reactants that actually allow for the chemical reaction to take place. Very little, if this whole curve is all the molecules, all the reactants, and only a small amount have enough activation energy for the reaction to take place, very few products going to be formed. But in our body, we have to make sure these reactions are going very quickly, all right, to keep up with the demands of the cell. In a catalyzed reaction, you can see we've added quite a bit more of the molecules that actually have the activation energy, enough activation energy, and so there would be more of the reaction taking place. Okay, you can think of kind of down here in a catalyzed reaction the amount of energy needed for the reactants. All right, here is the activation energy. This is the amount of energy these molecules need to make the reaction happen, for it to take place. Okay, so it's a wall preventing the reactants to turn into the products for the reaction to take place. Okay. This is non-catalyzed. This is without an enzyme. With an enzyme, you can see the wall is much smaller. The amount of energy that is needed for the molecules for the reaction to take place is much smaller, and therefore we can get more of the reactants to form the products. We can get more of the chemical reaction. So we speed up this process by lowering what we call this activation energy. Okay. So how does this take place? How do these enzymes operate to lower this activation energy? Remember, our enzymes are proteins, so they have that tertiary structure, and they have sites. They have these active sites, almost like little pockets in which the reactants can come and form a temporary bond with these enzymes. Okay, the old model was a lock and key model. Now it's believed that these active sites bind So as, say this is the enzyme and this is the active site, the molecule is coming in. As the reactant molecule is coming in, the enzyme is forming around it, doing this induced fit model. Okay. So you can imagine if we have these solutions running around, we have all these different solutes and we need to get A and B together and they're bouncing around. What if we have an enzyme that holds on to A and if B comes in, it holds on and we can get the reaction to take place. Okay, that's half of the part. We're also going to see that the enzymes torque the molecule to allow us to break bonds easier. Okay, but where the enzymes, where these reactants bind to or form this temporary bond is at the active sites. You can see we have to have this proper tertiary structure in our protein, otherwise the protein's not going to work. And this is why, because these active sites have to be specific because they're bonding to specific molecules. Okay, they're forming a temporary bond to specific molecules, so that active site has to have the proper amino acids around it so we get this proper interaction with the reactants. Okay, here's kind of the substrate coming in. All right, you can see that structure here rather fits for specific molecules. Okay, so these enzymes aren't just going to bind any molecules, put A, B, or you can take GF or any molecule. It's specific for these molecules. So they're specific for reactions or a class of molecules doing these reactions. Okay, so that's why we have to have all these genes, because we have to make enzymes for all these different reactions. These thousands of reactions that are taking place, we have to have specific proteins for them. One protein's not going to do the job of, say, 20 reactions. Okay, it is specific. But as they come in, there is this temporary bond that is formed between the reactants and the enzymes. But remember, the enzyme is not changed. Okay, and now we get coming out. Okay, this gets cleaved off of A, and that part that gets cleaved is bonded now to B there, and we get two new molecules. We just rearranged the bonds in these molecules. Okay, and we've made a chemical reaction take place. Okay, in your reactions, you usually see, right, the reactants written, and then the products on the other side with the arrow. Underneath the arrow, you'll see this is where the enzymes get listed. Okay, if the chemical reaction is catalyzed, you'll see the enzyme listed underneath the arrow. All right, so keep that in mind when you're looking at these chemical reactions. One of the ones I know you guys will see quite readily is carbonic anhydrase. And you imagine that converts converts water and CO2 to carbonic acid. So you can see, remember our carbonic acid, part of our buffer system. All right, so enzymes going to form temporary bonds, and what it does is it basically weakens the original bonds. Because remember, these bonds are strong. Those covalent bonds are really strong. So we have to have some energy put in so we can actually break the bonds, so then we can go and form new bonds. So that's part of it, is we have to break the bonds. We need energy to break the original bonds here. Here we got, right, we're breaking bonds in A. So we have to have energy, and what the enzyme does is lower the amount of energy we needed to break this bond here. And that makes it so we can speed up the reactions. Okay. So here's enzyme coming in, the substrate's breaking in, and we get products off. Again, it's temporary bonds. The example I like to look is, if I have a stick, say I have a stick and I'm here, and I want to break this stick. Right? hold on. I'll use my little birch trees here, right? Well, I'm not going to break it, but if I have to break this, I got to put a lot of force into breaking it. Okay? What if and I can't torque it, but what if I had it highly torqued already? Do I have to put as much energy in to break it? Okay, originally my activation energy was I had to put a good amount of force in to break it. Now if I already have it here, I only have to put a little bit of force, a little bit of activation energy to make that stick break. So that is basically what the enzyme is doing. As it's forming those temporary bonds, it's kind of almost, you can think of those molecules that's three-dimensional. It's torquing those molecules so those bonds, we can break those bonds easier and we can make the chemical reaction take place. All right, so that's how enzymes operate. That's how they lower the activation energy. They form these temporary bonds. All right, they basically weaken the bonds in the molecules, allowing us to break bonds. Okay, reducing the activation energy needed for the reaction. Now let's look at how, because we are going to see various enzymes throughout the semester, and I'm just going to focus on a few here. On how they're naming. What is the function? All right. So the naming are named after the function. All right. And some enzymes will have what the substrate name is, like lactic acid dehydrogenase. You see a dehydrogenase is an enzyme. So whenever you see an ACE, you're thinking enzyme. Okay, in this case, a dehydrogenase, D, remove hydrogen. We're removing a hydrogen from lactic acid. That is what that enzyme, that specific enzyme, does. Lactic acid dehydrogenase. It's not going to take an amino acid, some random amino acid, and remove a hydrogen. It's just going to do it for lactic acid. Okay, so that's one function, dehydrogenase. So if you see dehydrogenase, it's removing a hydrogen. Okay, I'm going to go synthase. We'll see this when we get into our cellular respiration. At the end, we will see a synthase when we're talking about forming our ATP. But basically, they catalyze dehydration reactions. Okay, hydrolysis. The two I really want to focus on. You guys will see these throughout the semester. We're going to see these a bit, but we're going to see phosphatase and kinases. Okay, phosphatase and kinase. A kinase adds a phosphate group. Okay, remember that phosphate group, that functional group. There's your phosphate in there and your two O's. Remember we saw that with ATP. That's the functional group. That these kinases, these enzymes, catalyze the adding of this functional group to various molecules. Many times it will be added to a protein. Okay, be added to a protein. The other is on the opposite end is a phosphatase. Phosphatase is going to remove a phosphate group from a molecule. Okay, again, many times we're going to see this in regards to proteins. Many times we'll see it in regards to enzymes. Okay, because this is going to be by adding a phosphate group or removing a phosphate group to a protein, we may be able to turn on or turn off an enzyme. Okay, because sometimes we want reactions catalyzing, sometimes we don't. So, okay, by having a phosphate group or removing a phosphate group from a certain protein, okay, we can turn. It's basically like a switch. Turn the protein on, turn the protein off. Okay. Don't get confused though, because sometimes proteins, when they have a phosphate group, they're inactive. And other proteins, when we add a phosphate group, that is what activates them. So it's not always kind of a protein with a phosphate group is turned on, right? And one without it is turned off. It varies between the proteins, whether a phosphate group added turns it on or off. Okay, but we're going to see this. This is called phosphorylation. Right? We will see this throughout the semester in turning on various proteins, mainly turning on key enzymes throughout the body. Right. So isozymes, they are basically serve the same function, okay, but these proteins are different in different organs. So even though this creatine kinase, and let's just look at the name, so we just went through the naming, it's a kinase. So what's it going to do? It's going to add a phosphate group. Okay, and what's it going to add it to? In this case, it's going to add it to the molecule creatine. Okay, and you can see in our brain, our heart, our skeletal muscle, we have an enzyme, creatine kinase 1, 2, and 3. We have enzymes that do the same function. The difference is the protein structure is different between CK1, CK2, and CK3. Okay. Why might this be important, since we are kind of headed towards the medical, or this is what this class is geared to, is what happens if somebody had a heart attack? Somebody had a heart attack, the heart cells are dying. Okay, they're not getting oxygen, they're dying. When they die, they break up and they release whatever in the contents of those cells. All right, if we had a heart attack, we could look for this specific protein. Okay, we have blood tests that can look for CK2, or another blood test can look for CK1 or CK3. So because these proteins are different, we can look for specific ones. In case if we suspect a heart attack, we would look for lots of this in the blood. Okay, we could also look for CK1 because maybe there was some maybe there was a stroke instead. The likelihood is with symptoms, you're going to look for one or the other. But in this case, CK1, if somebody had a stroke, then you're probably looking, or your suspected stroke, you would be looking for this isozyme. Okay, so medically, these isozymes are good because we can pick and look and kind of help confirm diagnosis of certain disorders, things that are going on. All right. So now let's look at kind of control of enzyme activity. Many enzymes, just the protein alone doesn't function as an enzyme. Many enzymes need what's called cofactors or coenzymes to make them operate. Others need to be phosphorylated or dephosphorylated to be able to operate. Also some properties that are happening, temperature, pH, those are all factors that affect enzyme activity. So that's what we're going to look at here. We'll see how they're controlled and so forth. And then at the end, we're going to look at these metabolic chains and control of these long enzyme pathways. Okay. So factors that are involved in enzyme activity, that have effect on enzyme activity. Okay. They would affect the way substrates converted a product. Some of these are temperature. One, pH. Okay, number of cofactors and coenzymes. We'll look at what these are. Again, also concentration of enzyme and substrate. Okay, if you have more enzyme but not much more substrate, you're going to have anything going. If you have concentration of enzyme and a large concentration of substrate, you're going to be making lots of products. Okay, and then there are some molecules that have stimulatory or inhibitory effects on formation of products. All right, so we'll look at all of these. We'll go through, look at all these and how they function and how they affect this enzyme activity. Okay, the first is temperature. All right, you can see on the X scale, temperature's increasing. And this is the rate of reaction. Okay, how much substrate is being formed into product. And as we increase temperature, the rate of reaction is increasing. Okay, remember the enzymes are here. If we got the molecules, our substrates are moving faster, there's going to be more and more likelihood that they're able to collide and mix together to get the reaction to take place. Okay, so this here, this increase in the rate of reaction is due to basically those substrates, okay, that are reactants that are their kinetic energy, their movement in that solution. Okay, what we see is at a certain temperature, okay, starts falling off. The rate of reaction starts falling off. And why is this? Is because remember these enzymes are proteins. They must have that three-dimensional structure. But with too much heat, what happens? Those proteins can start unraveling and denaturing, and if they don't have that proper active site, they can't catalyze the reaction, and that's what you see happening here. The reaction is not getting catalyzed by the enzyme anymore, and the rate of reaction. So more as we increase the temperature, more and more enzymes are denaturing, and we get a drop in the rate of reaction. Okay, so temperature. So you can see how we don't want to have extreme temperatures in our body. We can keep our, where do we keep it? Around 37 degrees Celsius. All right, if we have our enzymes, they have what's called optimal temperature. You can imagine ours, where would our optimal temperature, where the enzymes would work the best? And that would be roughly right around 37 degrees. Any temperatures varying too far off, the rate of reaction decreases. Okay. pH. Enzymes have what's called an optimal pH in which they operate. Okay, remember pH is the measure of what? Measure of our protons or our hydrogen ions. Okay. So you can see here are some enzymes we find in our body. And we say, oh, our blood is 7.4, 7.4. So shouldn't our enzymes be there? But these are enzymes of the digestive tract. Pepsin, salivary amylase, and trypsin. The key is where in the digestive tract these enzymes are working. Okay. Pepsin gets released in the stomach. From your anatomy days, hopefully you remember that pH is what? In the stomach is very, very low. Right? We secrete acid in the stomach, and so it wouldn't be ideal for pepsin to have an optimal pH of trypsin because it wouldn't operate. You can see it doesn't operate in that high pH. Right? Salivary amylase in the oral cavity is closer to neutral, so that's why it is good that the optimal pH is around neutral for salivary amylase. Right. And then trypsin is in the small intestines. If you remember, the pancreas secretes bicarbonate, and hopefully now you know bicarbonate, okay, buffers out. There's lots of bicarbonate. It will bind to this to form carbonic acid and would increase the pH. And so trypsin has a ideal pH, optimal pH, in the more basic range. Okay. For stuff that's in our blood, you can imagine what pH would you want to be close to? Optimal pH for any of the enzymes operating in our blood. Yeah. Right? Somewhere around 7.4. Not too far off of there, because that's where our blood pH, we try to keep that homeostatic right around 7.4. Okay. And you see, if we get too far away, you can see how pH would affect. If this was an enzyme in the blood, let's say over here, okay, if our blood pH went off too far, we could have drastically affect the rate of many reactions throughout the body. So many enzymes need other molecules to be able to function, to be able to catalyze reactions. One class of these molecules needed is called coenzymes. Okay, coenzymes are small molecules. They are biological organic molecules. All right, so they are these carbon. They are more complex than what we're going to talk about next, cofactors, which are just basically metal ions, but the same effect in that the coenzymes have to be available if there's a certain enzyme that requires the coenzyme. Okay, we will look at cellular respiration. We're going to look at the breakdown of glucose, which has many enzymes involved. And one of those enzymes needs this NAD molecule. All right, this coenzyme, it needs it in a certain form. If it is not available, that enzyme will not catalyze a reaction and essentially will shut down the breakdown of glucose at that step. Okay, many of these they use in transforming small molecules. We will be seeing many of the coenzymes that we look at through cellular respiration are going to be carrying or transporting hydrogen molecules. Okay, hydrogen ions. Right, so coenzymes, small molecules needed for the enzyme to operate. Right, similar is cofactors. Okay, cofactors help form the active site. Cofactors are not biological molecules, they are metal ions. You can see here's substrates, the enzyme without the cofactor bound, the active site is not available for the substrate. But once bound with cofactor, now we can bind to the active site and we can catalyze the reaction. Okay, so coenzyme cofactors needed for the reaction, for the enzyme to catalyze the reaction. Just one's a biological organic molecule, the others are metal ions, and the cofactors. Many enzymes can be modulated via allosterically. Allosterically means a molecule binds outside of the active site. It is not binding directly to the active site. Okay. It reversibly binds. So if it's for an allosteric inhibitor, all right, here's the enzyme, here's the active site. An allosteric inhibitor would bind somewhere, not in the active site, bind to the molecule. But you can see when the inhibitor is bound, the active sites change and now the enzyme cannot catalyze the reaction. Okay. Where on the other hand, an allosteric activation, okay, or activator, right, the enzyme's inactive, the activator binds allosterically, it is not near in the active site, and when the activator is bound, now the enzyme's turned on and we can catalyze the reaction. Okay, so the inhibitor or activator basically cause a conformational change to either decrease or increase the activity of the enzyme. Okay, allow the enzyme to catalyze its reaction. So this property of these enzymes and needing to have a proper active site to be able to catalyze the reactions can be utilized for, say, drug makers. We can compete, make competitive inhibitors that compete for, competitive meaning it's competing for the active site here. Okay, if a competitive inhibitor is in here, like we see down here, then the substrate can't bind to the active site and the reaction cannot be catalyzed. The reaction will be reduced. All right, so competitive meaning it competes for the active site, and it's inhibitor in that it's inhibiting the reaction from taking place. A classic example of this is your non-steroid anti-inflammatory, say like Tylenol. There is an enzyme that converts what's called arachidonic acid into an inflammatory molecule, inflammatory prostaglandin. And what Tylenol does is it is a competitive inhibitor for that site. It competes, and now we can't convert that molecule, that arachidonic acid, into an inflammatory molecule. So, and therefore an anti-inflammatory. Okay, so this is one way in pharmaceuticals in which we can compete for certain enzyme pathways, certain enzymes, to get a therapeutic effect. So besides modulators, enzymes, there are many enzymes have to be activated. They will be inactive, and then they have to be activated. Some forms of this is, some are formed as zymogens. Okay, the enzymes actually formed in the inactive form. The protein's formed, folded up, but until it actually gets cleaved or so forth, it's inactive. Classic example of this is enzymes of your digestive tract. You have zymogens, because zymogens, these digestive enzymes, are going to be there. They're going to break down proteins that we eat. They're going to break down carbohydrates that we eat, lipids that we eat. We don't want these enzymes active in our cells, because our cells are made up of what? A bunch of carbohydrates, proteins, lipids. Our cells are made up of all these molecules, so we don't want these active enzymes in our cells. Okay, our cells make these enzymes, but they make them as zymogens, these inactive form, and then when they get secreted in the digestive tract, they get activated. Okay, pepsin is one of those. Pepsin is called pepsinogen, is its inactive form, and then when it goes into the digestive tract, it gets cleaved and becomes pepsin, and now it can digest proteins for us that we eat. Okay, that's one way of activation. I did speak of phosphorylation and dephosphorylation. That is adding, all right, adding or removing that phosphate group, that phosphate functional group. So adding one of these functional groups to a protein, or removing one of these phosphate groups from a protein. Okay, this can turn the protein on or off. And again, I discussed earlier, is some proteins are active phosphorylated and some are active dephosphorylated. Just depends on the protein. Okay, but we can turn them on and off by adding a phosphate or removing a phosphate from the enzyme. Okay, hopefully going back, what kind of enzymes, what's the name of the enzyme that adds a phosphate group? A kinase adds a phosphate group. I'm not going to spell it, but the dephosphorylation is a phosphatase. A phosphatase removes the phosphate group. Okay, so we have to have, if we're going to add an enzyme, we have to have a kinase that basically adds a phosphate group to another enzyme. Okay. Also, enzyme inhibition be controlled by turnover, okay, in which there's enzymes that degrade it. If there's more of these enzymes that degrade it, we can turn over the enzyme more regularly. So these are ways in which we control the activation. You ask why maybe we phosphorylate or dephosphorylate? Why don't we just make more enzyme or make less enzyme? Key is remember our protein synthesis. Our protein synthesis, pretty drawn out steps. We got to transcribe, we got to translate. This takes time. All right, it doesn't happen instantaneously when we do our protein synthesis. So what the body does is we can make these enzymes, these proteins, and now we can just turn them off or on. Okay, that's a better, easier, quicker, very more rapid way of regulating the enzyme. All right, if we had to make them or degrade them, that would take much longer. And remember, these chemical reactions are taking place very quickly in the body. The cells need them to take place very quickly. So this is a good way in which to have very quick regulation on and off of these different enzymes. So another factor regulating the activity of enzyme is substrate concentration. I don't have also here, but also enzyme concentration. Right? As long as there's substrate available, if we added more enzyme, also we could speed up the reaction. But let's look at substrate concentration. Okay, we can add more substrate. The key is you look at the rate of reaction here. Reaction rate's here on the Y. Substrate concentration is here on the X. You'll see as we increase substrate, the rate of reaction does increase, but what happens is, all right, there's a point where there's a saturation of the enzyme. Okay, remember we need the enzyme to catalyze the reaction. Enzymes have active sites, so they have to bind the substrate. What happens if we have so much that, okay, the enzyme's bound by the substrate? All the rest of the enzymes can't be bound until this reaction gets catalyzed, and then the new molecule can come in. So we get this point where the enzymes are saturated, right? And so that's why we get this leveling off. Even though substrate concentrations are going up, the rate of reaction, okay, levels off because there's only so much enzyme in which to catalyze the reaction. Okay, if we say we added enzyme, we increasing the substrate, and here we added more enzyme, we'd have more enzyme, and we could even increase the rate of reaction even further. Okay, there's a combination. You got to have substrate and enzyme. At a certain point, substrate's too much, enzyme's only here. All right, the reaction rate is going to level off due to saturation. So as we've seen, some reactions go in both directions, and some enzymes can catalyze the reactions either forward or backwards. Okay, depending. And again, though, the same laws apply in the law of max action, in that which way the reaction's driven has to do with the concentrations of the product and the substrate. We reach this equilibrium in the equation, and then if we add something, it's going to get heavier, and we're going to get the teeter-tot. Okay. Classic is carbonic anhydrase. I've already mentioned this one, but that is version of H2O and CO2 to carbonic acid. This one should start looking familiar to you. But let's say if we hold our, look at this, we get this chemical equilibrium. I'm going to do a little more chemistry just so I draw the effect, but we get this chemical equilibrium. What if we hold our breath, though? Okay, if we hold our breath, what do we retain? We retain CO2 in the blood. So that would make our equation heavier on that side. How do we get it back? All right, equation would be drawn this way towards carbonic acid to bring us, that would push that side heavier, and we can get back to our chemical equilibrium. Okay, or reaction equilibrium. Okay, but same thing, same principle, law of mass action. It's just that the reaction is driven or catalyzed by a reaction or an enzyme in both directions. Carbonic anhydrase will catalyze carbonic acid into CO2 and H2O, but it can also form carbonic acid from H2O and CO2. So where we're driving with this, or as we head towards the other part of chapter 4, and that's kind of why enzymes are brought here, is we're going to see these enzymes exist in these metabolic pathways. Chapter 4 deals with cellular respiration as we break down glucose. And in the first step, we break down, in glycolysis, we break down glucose into what's called pyruvate. Here's glucose, we break it down to pyruvate. But this isn't done in just one step. There are multiple steps along the way. In each step, there is an enzyme that drives the reaction, and then there are intermediate molecules. Okay, so we're not going glucose to pyruvate, right? We're going to see we go glucose 6-phosphate is the first step, and there are multiple 9 steps to this glycolysis process, and each step is catalyzed by a reaction. In each reaction there is a product or an intermediate. Okay, there's not total 9 steps here, but if we had glucose here, the end we would end up with pyruvate, and we would have multiple enzymes here catalyzing reactions. So there are these pathways. They lie in this chain. Okay, you begin with an initial substrate and end up with the final product, but again, it's not jumping from here to here. There are all these enzymatic steps along the way and intermediate products along the way. Okay, in these metabolic pathways. Most metabolic pathways are not linear, meaning that they, as we saw on the first slide, they all go from A to just one product. Many of metabolic pathways are branched. You start with A, you get to B. Now B can go to C. C now can go one of two directions. Either make D, or you can see the little D knot here. Okay. These are different enzymes. This is a different enzyme from enzyme 3 knot. I wish they would have made this a little bit more clear, but now, depending on which branch we took, we can end up with different products. Okay, so starting with one substrate, we can end up, and we can even have a third branch or another branch down here and end up with 3 different products from the initial substrate. Many pathways have what we call end product inhibition. Remember allosteric inhibition. A molecule can bind to an enzyme and it can inhibit the activity of the enzyme. Okay, this is the process and where we're headed towards for our cellular respiration. This is called glycolysis here. You don't need to know it right now, but this is one of those metabolic pathways we will be looking at. Here is glycolysis. We start with glucose, and we end up here with 2 molecules of pyruvate at the end. Okay. So what end product inhibition means is if glucose, basically this glucose is being broken down by glycolysis to ultimately form ATP. But what if we've already met our energy needs? We'll see we don't make ATP big stores of it, because if we made big stores of it, it actually will convert back to ADP, and basically we wasted all this energy making ATP. So if we already have our stores made, we don't need to be breaking down glucose through glycolysis. Okay, if we've done a lot of glycolysis, our energies are met for our cells, we have lots of ATP available. Okay. One way to shut this down is we can shut off this process of glycolysis. We don't need to break down glucose. And you can see some of the products that are in further down in this pathway come back to inhibit some of these earlier enzymes, allosteric inhibitors. So if we inhibit this enzyme, we can't take fructose 6-phosphate to glyceraldehyde, or sorry, fructose 1,6-bisphosphate. We can't make this reaction, which is key because if we have our energy stores made, we don't need to do glycolysis, and it makes sense that an end product would come back and inhibit glycolysis because we don't need to keep doing glycolysis if we already have enough ATP. Okay, we're going to look into this in more detail when we get into the glycolysis process. We'll actually talk about this one enzyme here. But just realize in many of these pathways, some product at the end of the pathway can come back and inhibit one of the earlier enzymes, and that would shut down the pathway or slow down what we call the flux through these pathways. Right? In this case, one of the later products is coming back to inhibit this enzyme, and therefore it would inhibit this step in the pathway and inhibit the overall metabolic pathway. So here's kind of better rendition of it is we go through A to B to C, and say we're going in this direction. All right, this product has accumulated, we got lots of it. Now, if this product can be an allosteric inhibitor to enzyme 3, we can go back and inhibit this enzyme, and that shuts the pathway from going in this direction because we have enough of this product. We don't need to be making more of it. And so this allows us control of these metabolic pathways. Now, C would be, if this pathway isn't inhibited, would go down this pathway to make our F. Okay, so this is how we get some of this regulation, this control. We're going to look at this. This is just kind of introducing you to the metabolic pathways. Since once we get into cellular respiration, we're going to be looking at these pathways in more detail. In these pathways, there can be inborn errors, or basically mutations. What happens if one of those enzymes is not functional? Okay, let's look at it if it's at a branch point in one of these pathways, one of those enzymes in the branch. Say, in this case, enzyme 3. There was a mutation. This enzyme's not functional. All right, we can go to A to B and get to C. Problem is if we can't get here, we can't get down to the final product of F. All right, and in a normal individual, this is set up so we can send some of C this way, some of C this way, because we only need so much of F naught. All right, in this way, in these errors, we can get disorders if this is an important product. Okay, if this enzyme is bad, we can't make F. All right, and we can get a disease from, say, a lack of F. If we can't make F, we can't make that product. We could get a disease possibly from, depending on what product we're talking about, we can get a disease from F. How about this way? There used to be flux movement through this direction and this direction. Some of C went this way, some of C that went this way. Now all of C is going down this pathway, and so we can have an overproduction of F naught, because some of it used to be turned towards F, but now it's only F naught. Okay, so we could get a disease in where there is accumulation or too much of F naught. A classic example of this is overproduction, right? Is phenylketonuria. And all babies in the United States, when they are born, they get tested for PKU. Okay, you can see here is that branch. We got phenylalanine. Gets branched to make this way and branched to be converted to tyrosine. In fetal ketonuria, this phenylalanine hydroxylase is not functional. All right, phenylalanine typically goes this direction and this direction, and so we get a good amount of both of these products in both directions. But now this isn't being converted to tyrosine, so all of phenylalanine gets shunted into this pathway, all right? And phenylpyruvate, phenylacetate, accumulation of this or too much of this can actually cause brain damage and seizures. Okay, the key is where do we get this phenylalanine? Is we get it through the diet chiefly. So if we limit the amount of this in the diet, all right, we can prevent a substantial overaccumulation of this and prevent the brain damage and seizures. So that is why these babies get tested, is because there is a way to prevent it. If we don't put this in the diet, then we won't have those seizures and we can get normal development. Right, so this finishes up with our enzymes. What we're going to head towards is we're going to head towards cellular respiration. You'll see why this comes into effect, why we kind of touch on base on enzymes and why your books have enzymes in that chapter, because there will be these metabolic pathways with all these enzymes and these steps and this regulation through those steps. All right.